Hey, Assalamu alaikum. In this video, we will understand about the fatty acid beta oxidation. So the beta oxidation of fat in this video we will understand. So let's understand about all these things. Please watch the whole video. So metabolism and ATP production we will understand in this video. So for example, the basically the fatty person whenever will stop the eating as a fasting. So the fasting will not allow to carbohydrate metabolism and ketogenic diet or exercise also. So basically, for short, the fasting basically inhibit the glucose metabolism while trigger the fat breakdown. So adipose tissue will be breakdown lipolysis to produce triacylglycerol to the fatty acid and glycerol breakdown. So in this way here is you can see the beta cell of pancreas will stop the production of insulin due to the low level of the glucose due to the fasting in the blood is known as the hypoglycemia, while the glucagon will increase with the decrease of the insulin. So in this way the insulin deficiency will trigger the hypothalamus to trigger the growth hormone, which that is the alternate hormone for the growth and the maintenance, and basically it is important. And in this way the growth hormone will target to the adipose tissue, and glucagon will lipolysis to produce free fatty acid and glycerol in the blood. So this free fatty acid go into the liver to produce ketones it is known as the ketogenesis, and this ketogenesis produce the ketone, which that is the alternate for glucose. So the glucose will decrease while the ketones will increase for the giving the energy to the brain and due to the electron transport chain and the Krebs cycle. So here the free fatty acid will be in the presence of enzyme acetyl-CoA synthetase enzyme to convert the free fatty acid in the outer membrane of the mitochondria to fatty acetyl-CoA. This carnitine palmitoyl transferase enzyme, which that palmitic acid, for example, but we are just understand that generally palmitic acid will enter due to the CPT1 with combined with the carnitine. So carnitine and fatty acetyl-CoA combine together to transport through a carnitine palmitoyl transferase 1 enzyme. So in this way the acyl carnitine will enter into the outer mitochondrial matrix. So in this way the inner mitochondrial matrix require the carnitine palmitoyl transferase enzyme 2. This carnitine palmitoyl transferase enzyme 2, which that allow to acyl carnitine enter into the inner mitochondrial matrix. So remember about that things. This is the transportation, and this is the carnitine shuttle mechanism. In this the carnitine will again back toward the outer membrane, and in this way the cycle will begin. And this is the shuttle mechanism because the carnitine is the transporter of the fatty acetyl-CoA after the formation via acetyl-CoA synthetase enzyme. So in this way the acyl carnitine will enter due to the CPT2 enzyme, and this transportation will enter into the inner mitochondrial matrix, and this fatty acetyl-CoA will detach with the carnitine via enzyme, and on the other hand the beta oxidation will occur from the fatty acetyl-CoA to produce fatty acetyl-CoA and the acetyl-CoA. These two carbon will be separated from the beta carbon, and in this way the breakdown of the beta carbon to release the acetyl-CoA, which that is the 2 carbon containing, while the rest of the carbon which that remain in the form of fatty acetyl-CoA, and that again cycle will begin. While the carnitine, which that carnitine translocase enzyme used to translocation in the outer mitochondria, so outside of the mitochondria, will again, and that again pick up the fatty acetyl-CoA to again cycle. This is known as the carnitine shuttle mechanism. Anyhow, let's understand about this basically the inner mitochondrial matrix. This inner mitochondrial matrix, basically the fatty acetyl-CoA will be in the form of different enzyme which that lead to beta oxidation. The beta carbon will be detached and the fatty acetyl-CoA will be left behind, while it depend on the number of carbons which they recycle again to use more acetyl-CoA, and more acetyl-CoA will enter into the Krebs cycle to produce the ATP more and NADH and FADH2. This NADH and FADH2 will produce, and in this way the ATP production will be more as compared to the glucose, because the more and more acetyl-CoA will be formed as compared to the glucose. Basically the glucose produce 2 acetyl-CoA due to the glycolysis to produce pyruvate, and the pyruvate will enter into the mitochondria, and acetyl-CoA, 2 acetyl-CoA formation will occur. So anyhow, this is the major concept. So in this way we should understand the beta oxidation of fatty acid with detail. So there is the carbon dioxide and water and ATP produced, but important thing you should need to understand, this is the aerobic respiration require the oxygen in the electron transport chain of the mitochondria to produce ATP, and require the oxygen for the accepting the electron to prevention of the free radical. But the free radical prevention different, but this is the ketogenesis also formed due to the acetyl-CoA to convert the 3-hydroxyacyl-CoA, and this basically will lead to ketones body production due to the fasting. Anyhow, let's understand about the beta oxidation of fatty acid. In this way the fatty acyl-CoA will enter in the pathway of the beta oxidation. Let's understand about that. So the fatty acyl-CoA basically contain a number of carbon, and the terminal region which that contain a beta and alpha carbon, which that attach with the coenzyme. So in this way I hope you make sense about that. I will mention the red color pen to understand. So here is the hydrogen, and this is the single bond, and alpha and beta carbon are present. So this is the acetyl group. Basically formation will occur after the breakdown. So anyhow, this let's understand about the FAD plus will be reduced in the form of FADH2. So the 2 hydrogen will be removed in the presence of the enzyme is known as the acyl-CoA dehydrogenase enzyme. This acyl-CoA dehydrogenase enzyme will accept the proton via a FAD plus into the FADH2, while this is the oxidation of fatty acyl-CoA. This is the oxidation process because the removing of the proton, the 2 proton, in this the carbon will become a double bond. This double bond carbon is known as the trans delta 2 enoyl CoA formation. This basically the hydroxyl group will attach with the beta carbon, and this is the basically the trans state of the enoyl CoA, and the carbon alpha which that accept the proton due to the water hydration is known as the hydratase enoyl CoA hydratase enzyme used by the presence of the water. So the water will split as a hydration. So the water will split. This is the hydration phase. This water will split into the hydroxyl group and the proton. That hydroxyl group and proton will accept via a beta carbon and alpha carbon to again pi bond reverse, because this pi bond is unstable. So again very quickly to form hydroxyl group with a beta carbon, and alpha carbon contain proton, the hydrogen. In this way, this basically will lead to 3-L-hydroxyacyl-CoA formation. These 3-L levohydroxy acyl-CoA formation will occur. The coenzyme will be attached, remain attached. So in this way, this formation basically will form a beta keto-acyl-CoA. This beta keto-acyl-CoA due to the basically the removing of the proton, while the oxygen will become a double bond to form a keto group attached. That is why it is known as the beta keto-acyl-CoA. So first is the 3-levo-hydroxyacyl-CoA formation, and this 3-hydroxyacyl-CoA dehydrogenase enzyme will dehydrogenation of this 3-L-hydroxyacyl-CoA and reduced NAD plus in the form of NADH. And that is why it is known as the dehydrogenation. So this dehydrogenase enzyme used to accept the proton by a NAD plus, while dehydrogenation will occur in the 3-L-hydroxyacyl-CoA. This 3-hydroxyacyl-CoA basically will remove the hydrogen. So in this way it will become a it will become a beta keto acyl CoA. So remember, the beta keto acyl CoA contain a keto group. That is why it is known as the beta keto acyl, because the beta carbon contain a keto group. That is why it is known as the beta keto acyl CoA. And in this way, the further coenzyme A will again bind in the form of reduced, which that beta ketothiolase enzyme use to basically donate the proton to the beta ketoacyl CoA, and in this the proton will enter, and the proton basically bind with the carbon beta, not carbon beta but the carbon alpha. So the carbon alpha contain a addition of the hydrogen to become a acetyl-CoA due to the CH3 C double bond O. So you can understand as well. So here is the beta ketothiolase enzyme used, and in the presence of the HS coenzyme A, which that is the reduced to oxidize to donate the proton to the alpha carbon, and in this the CH3 formation will occur, while on the other hand the beta carbon basically contain a double bond. So in this way this will lead to again formation of the acyl CoA fatty acyl CoA formation. This fatty acyl CoA again will be reused for the again beta oxidation until the complete oxidation of fat will occur. So the beta oxidation basically more production of energy, and fasting produces ATP more due to the beta oxidation of fat, which that is the reserve in our body. So the ketones best than the sugar because it will produce more and more ATP easily and efficiently, like the jet fuel. Starvation good than the eating, and the fat reduction basically fat lose easily. So I hope you make sense about that. So the more energy production, the fasting produce ATP more, ketones best than sugar, and the starvation good than eating, and fat reduction will increase. So in this way you can lose your fat during the fasting and exercise. So I hope you make sense about that. The acetyl CoA will be occur due to the beta oxidation, and fatty acyl CoA again used for the beta oxidation. In this way, the acetyl CoA used for the Krebs cycle to produce energy, and that is more than the sugar. I hope you make sense about that. Please make sure to subscribe, like, and share. Bye.